Good evening, everyone. This is Muskan Gaur, and on behalf of my whole team, welcomes you all to today's session on Pulse of Plants. I would like to thank our current sponsors, especially Reliance, competition partner, Hindustan Platinum, our title sponsor, J Chemicals, presented by sponsor, Heikel, association partner, Alkyl Amines, industry partner, and WAC, our education partner. And last but not the least, all our attendees for joining this event. I hope you all enjoy and learn from today's event. I would also like to welcome our speaker for who took from their busy schedule to enlighten us, Professor Abraham Stroock, who is a professor and the director of the Smith School of Chemical and Biomolecular Engineering at Cornell University. Professor Abraham's research relates to engineering microchemical processes with an emphasis on transport phenomena, thermodynamics, and physiology. He obtained his BA in physics from Cornell in 1995 and his PhD in chemical physics in 2002 from Harvard University. So we welcome you all, Professor. Now I'll hand over to you and we are good to go. Thank you.
Professor, I think you are muted.
Thank you. Thank you all. Thank you, Muskan. Thank you, Niragan. Hello, everyone. It's a pleasure to be here and to be closing out your Azeotropy 2021. I am very sorry that we're not all together on your campus. So today I'm going to speak to you about a topic that it may seem a little bit unfamiliar, and I think it is unusual within chemical engineering to be looking at plant life. But I'm going to try to give you a sense of how it's an important topic and a topic for which we are very well trained as chemical engineers. So I am here's some shared plant life on our two campuses. On the left here is a garden on the campus of Cornell, and I actually live just outside the photo. Where I'm sitting right now is just outside the photo. To be honest, in the springtime here right now, the garden does not look like this here in North America. But it's plants are one of the universal characteristics of our planet. And then below I'm showing where we Mumbai and Ithaca on the map. And again, very much saddened that we're all living under COVID and that I was unable to join you in Mumbai today. As I was telling the organizers, I last night, as a small consolation, I watched YouTube videos of dosas being made on the streets of Mumbai, and it made me even sadder and very hungry. So, so just to note, at any moment, please put questions into the chat. Kriti told me that she would shut me down and force me to answer questions, and I'm very pleased to answer questions at any time. So, as parts of motivation, what motivates me heavily to study plant life is that it's beautiful. It's beautiful, and in an important sense, it captures the physics and chemistry and biophysics that is of interest in chemical engineering. And like I said, I think chemical engineers are unusually well prepared, unusually well trained to understand and analyze and influence how we interact with plant life. And so if this topic becomes of interest to you, this is a review that we wrote for a community like ours, an engineering community, about plant physiology. So plants are also critical in our planetary system. This is showing the evolution through years, each year, of the plant coverage on our planet, which covers about 72% of the land surface. Of that 72%, well over 10% is under direct human control in the form of agriculture. And so in this sense, the plants are active, critical boundary condition on our planet's atmosphere and hugely important in controlling our climate. Measured another way, plants are by far the largest form of life by mass on our planet, 80% of biomass. Here, this little pink circle is humans, and even bacteria and microorganisms in general are dwarfed by the scale of plant life. One way to think about this boundary condition, this active living boundary condition on our atmosphere, is to look at this famous, infamous curve where a CO2 concentration is evolving through the years. And as we all now have grown to appreciate, the black data here is the trend line that is certainly under human influence. The blue line, on the other hand, is the month-by-month data. And what we see is that each year we have massive changes in CO2 concentrations, rapid changes that are essentially controlled by plant life. And here again, this is this rapid flux of carbon into and out of the atmosphere is heavily under our control. Some 10-20% of it is directly under our control in the form of agriculture, and all the rest of it is indirectly under control in how we influence our natural ecosystems. Plants are critical in that agriculture that we pursue is by far the largest consumer of human consumer of fresh water. 70% of all human extraction of fresh water goes towards agriculture, and by some measure, almost half of it is wasted, meaning it doesn't get into productive form within plants. Plants feed us, and their ability to feed us is under strain. We have a growing population towards 10 billion, and across economies, across agricultural systems, from very productive agricultural systems to growing productive agricultural systems, there is potentially a real plateauing in our ability to create more calories per hectare. In an important sense, plants are neglected, and when I say neglected, they're neglected by us, by engineers. Whereas here on the right, there's been huge investments and intellectual and financial into creating technologies to interact with mammals, and in particular humans. We lack even the most basic crucial tools to interact with plant life. I show at the top a hypodermic needle. Hypodermic needle is a foundation of modern medicine, and even that simplest of tool for intervention to get into the internal workings of the organism, we lack in plants, and that's one of the things I'm going to try to give you an appreciation for, why plants are actually present particular challenges to build the tools to get to internal biology. And on the left, I have a list of things we'd like to measure, things we'd like to be able to measure in real time. I'm going to speak in particular about water stress, but if you're looking for an open area for engagement with biology as an engineer, essentially it's all open for invention and innovation and discovery. So the vision I'm going to develop, and I heard from Nirajan that you had a discussion of Industry 4.0. There's a maturing vision and implementation in our traditional industries of an Internet of Things, network sensing and actuation embedded throughout, for example, a chemical manufacturing process. And the ingredients of the Internet of Things and Industry 4.0 is both this ability to engage at local scales within unit operations in a process, inform continuously models, what can be called digital twins of those processes and of the integration of those processes, and to come back with control and optimization. And I point to the opportunity the red line is an opportunity for learning and discovery. And at this stage, what I'm going to tell you about is really trying to build out this red line in a new context, in the context of plants. Here, a vision that I'm part of pursuing at Cornell, we're calling an internet of living things, where we'd like to engage plant life, plant systems, the organism, the plant organism, and the associated organisms like the microorganisms in the soil, and engage them in this same sort of closed loop of data and from individuals, from inside individuals, into multi-scale biological understanding, so that we can control plant systems principally in agriculture, but also in natural contexts, in sophisticated ways that are informed by the biology, from the genetics through the physiology through to the population scale of the landscape and the ecosystem. So within this, my engagement is at the scale of building the nanotechnology, microtechnology, biotechnology that allows the internal biology of the plant to be expressed outward and digitized, and allow us to first understand, build models of essentially the unit operations of plant life, so that we can work towards a place where where agriculture can, parts of agriculture at least, can become engaged in the Industry 4.0 level control and optimization. So the unit operation I'm going to focus on is water transport, in particular from the soil through the plant, maintaining hydration under sustaining the biochemical processes of a living organism, and then escaping out into the atmosphere. And so there's a very complicated little sketch here, and I'm not asking you to look carefully at it, but what it's showing is the whole richness from the atmosphere of turbulent transport processes in the atmospheric boundary layer, radiative heat transfer in exchange with the sun. As we move into the leaf, we have living tissues under biological control. Here, this is where the plant actively controls its role as the boundary condition on the atmosphere. Meters squared per plant of exchange area for both radiation and mass. Into the tissues, it becomes very much a chemical engineering transport problem. Viscous flows through porous media. Into the soil is a complex porous medium, multi-phase, and so there's a richness throughout of thermodynamics and transport coupled to biology and the environment. And that's what really attracts me strongly intellectually to this problem. So water controls everything. If you've ever tried to keep a plant alive in your apartment or home, you know that it controls growth, it controls the yield of crops, it controls in subtler ways quality and also disease. But despite its central place in plant life, in agriculture, it basically remains open loop. We actually, thanks to companies like Jane from India, there's millions and millions of acres of controlled delivery of water, but essentially no robust way to close the loop on the delivery of water based on the state and needs of the plant. And so this is where I mentioned a factor of 2, that if we did control irrigation in a precise way informed by the biology, we could save 40+% of the water we use that we are currently delivering. So that is a target for this Industry 4.0 of agriculture. So the trouble is, water stresses hard to measure. We don't have that hypodermic needle to access the internal state of anything inside of a plant on a regular basis. And so our vision on the left is stymied by the fact that right now we have to typically take a piece of tissue off of the plant, put it into this pressure chamber, squeeze it, watch it by eye to assess the status of water stress in a plant. And so clearly we're not going to build an internet of living things based on a manual tool like this. So why is it hard to access the state of water in a plant? Why is it hard to enter a plant at all? What is the fragility and challenge? Well, so here I'm taking a grand example of a plant organism, a California sequoia, and pointing out the local porous medium structure that is the vascular system, the xylem of the plant, through which water goes from soil root to the canopy or leaves. And the point is that there is no pump in a plant. There is no pump, and in fact, water is held from the top. And so water is serving as what I call here a liquid rope. It is pulled from the top, pulled out of the soil based on stress carried along its length from the top to the bottom. And like a rope, like any rope, it's prone to break. Okay, and so this is an odd scenario, unfamiliar scenario, that liquid in plants is almost always in a state of tension or negative pressure. And I will try to justify this briefly. And one of the ways we have advanced in this, and is often a good way to take a complex system and make it analyzable scientifically and in terms of engineering and engineering design, is to take a simplest version, extract the rudiments of the system. And so here I'm taking this porous medium, which is representative of almost all of the plant tissue, and making the simplest version of what it would be ideally. And ideally, it's what we'd call a bidispersed porous medium. It's got these vessel structure that is on the macroscopic scale, tens of microns, so a capillary scale on the micron scale. But throughout, it also has a nanoscopic pore scale, that separates it from the outside world in the roots and separates it from the outside world in the leaves. And I'll try to, so the microscale is kind of self-evident. It is to improve conductance so that there's not excessive pressure drop along the system. The nanoscale, I'll try to justify, its origins are hinted at by these curved menisci. It is a critical length scale in the design of plant tissues, and in the end, it's going to be the design of systems we've used to interact with plant systems. So here's a scenario where we have this minimalist plant interacting as plants do with an outside environment. And what's important about the outside environment from the point of view of water is the activity or relative humidity, the vapor pressure over the local saturation vapor pressure. And except during monsoon, except when it's raining, that number is less than 1. And we would like to understand the transport processes in the condensed phase within the tissue. And so we're going to ask, what is the liquid pressure, given that we have locally equilibrium between the exterior boundary of the plant and the outside world, which is unsaturated. So this is a little bit of equations in simple thermodynamics, but bringing us into an unfamiliar regime. So we say, what is equilibrium with respect to mass transfer? It's equality of chemical potential across these phase boundary. We have the vapor phase chemical potential equal to the liquid phase chemical potential. If we take the simplest representations of chemical potential for the liquid phase, it's really only depends on the pressure relative to the saturation pressure, and this molar volume would come over, and that's an equation of state for the liquid. On the vapor side, we take an ideal gas law, and we have RT times the logarithm of the activity or relative humidity. So this says that for this local equilibrium to occur, there should be a change in the liquid pressure relative to the standard state pressure, reference pressure. This must be balanced by something, and this brings in the curved meniscus here with a contact angle theta with this small scale pore of radius RP. And this is the surface tension of the liquid with its vapor or with air, and a cosine of the contact angle. And we find this same pressure difference must be sustained by the capillary pressure across this curved meniscus. So together, these are starting to tell us in terms of physical chemistry and chemical engineering concepts, what is actually a good definition of stress, of water stress within a plant system. And it's got a name, it's called water potential. It's a recasting of the chemical potential. And it's defined in pressure units as the actual pressure in the liquid in the condensed phase minus the standard state pressure. And it's equal to this RT times the logarithm of the activity divided by the molar volume. And this has units of pressure. And this, for this system to work, the meniscus must remain stuck at the interface. And so this has to stay, less than in magnitude than the maximum capillary stress that can be created by this small scale pore. And so this is where this nanoscale pore becomes important. Why do I say nano? Well, if we take just 10% drop from saturation, so this is an activity that's equivalent to 90% relative humidity, which feels like a very humid day, already we need 14 megapascals, which is 140 atmospheres or bars of stress. That is what is implied in the liquid phase if it's coming to equilibrium with a vapor phase. It's really quite humid still. To maintain that with a capillary stress requires pore scales that are no larger than 10 nanometers in diameter. 50% relative humidity is not such an extremely dry day. That leads to 90 megapascals or 900 atmospheres of stress, and that would require 1 nanometer scale pores. And I'll point out again that this stress difference is negative, and this liquid pressure is negative, absolute negative. And so the tiny bit of dryness in our world leads to enormous potential stresses in our plant life. And that is crucial. So here, Priti hasn't stopped me yet, but I'm going to stop and ask you a question. This is a quiz. So which of these species experiences the largest water stress in its vessels? And I'll wait for you to put some answers into the chat. And if you don't put them in, then Nirava and Kriti and the other organizers are going to have to give me some answers. So we have one, I guess, for the sequoia. Are there any other coming through? Sequoia, 2 for the sequoia. Mangrove. I think you have mangrove maybe right around Mumbai. So sequoia, mangrove. Nobody likes this little bush as an option. So why sequoia? Anybody say why Sequoia? It's the largest. It's the largest. And we think, you know, we know that a column of liquid held from the top is experiencing a pressure drop of rho times g times h, rho the density times gravity times the height. And that seems like a pretty big stress. And then there's the viscous stress of pulling liquid through all those little pipes up the plant. But actually, thermodynamics trumps mechanics here. Sequoias and other large species tend to live in wet environments where this thermodynamic stress that I just described to you is minimal, and all they have to overcome is the dynamic stress of gravity and viscous pressure drop. It's actually the plants that fight thermodynamics that are the larger, that experience the larger stress. Mangrove are fighting the osmotic stress of the of brine, of the ocean water. Day and night, a mangrove must maintain a tug of war between nearly pure water in its vessels and 3.5 megapascals or 35 bars of osmotic pressure in the ocean. This sad-looking little bush has an even more brutal tug of war going on, living in almost desert conditions, holding on to its water as a stress, with a stress against the drying of the environment, the dryness of the environment. So good. Okay, now one more quiz. Are negative pressures in liquids even physically allowable? Does this bother anyone? This idea of absolute negative pressures in liquids? Did anybody's chemistry or physics teacher ever tell them vacuum can't pull? Some potentially confusing statement like that? Any thoughts on this? No? No one's bothered? Maybe in the case of an onion plant. I'm not sure I understand. I'm not sure I understand that reflection, but why onion? Any other thoughts? Negative pressure is okay. Negative pressure will cause the liquid to vaporize. Yes. Negative pressure should cause the liquid to vaporize. So, thank you, Aditya. It should, and it will eventually. And so, though, it turns out that all condensable liquids can sustain some stress. And so what I'm showing here is our typical picture of PV of an isotherm. This is the liquid branch. This is the coexistence line or binodal. And this is the vapor branch of a condensable fluid. Okay. What we don't usually see in our textbooks is the whole cubic equation of state isotherm, which shows that the liquid branch actually goes right through the binodal and through zero and keeps going down until it reaches a minimum, which is called the spinodal. This is the spinodal of the liquid. This is all thermodynamically possible. And when the vapor, if we extend the vapor out to unsaturated conditions, larger molar volume, the liquid will try to stay in equilibrium with it by continuing to follow the liquid branch down. And it can do this in a so-called metastable state down to what is for water very negative pressures, potentially well over 1,000 atmospheres of tension, until it becomes absolutely unstable, the dashed line of the absolutely unstable states. And so this is what is called metastability. It's thermodynamically unstable, but it's kinetically stabilized by the energy barrier it takes to create that first bit of vapor within the bulk liquid phase. And so this is the exotic regime in which plants are operating. So are there questions from the audience?
So before you proceed, I think I could take up a few questions. So the first question that we have is, sir, what do you think about the hydroponics and aeroponics in agriculture?
So that's a good question. Hydroponics and aeroponics minimize concerns about this water stress. They give you perhaps the best control of the delivery of water to the plant. And importantly, what I will show you in the coming slides is that the soil presents an enormous complexity when we try to model the plant system. So by going to aerobonics and hydroponics when possible, now of course that's truly turning plants into a factory kind of setting. You have huge advantages in terms of the types of control that I am pointing to. So it's a good question. Of course, it's not on a short term that we are going to be able to abandon soil as the host for most of our agriculture. But good question.
Thank you so much, Professor. So the next question that we have is, how does the increase in the concentration of carbon dioxide in the atmosphere lead to the change in water potential of the water vapor present in the atmosphere?
So it turns out that the exchange of water and CO2 are intimately coupled in the way the plant manages its engagement with the atmosphere. And so availability of CO2 and sunlight drives the plant to open itself so that it can feed photosynthesis. Inevitably, though, and I'll illustrate this in the coming slides, that leads to the loss of water into the dry atmosphere. And so plants are stuck with a compromise between their need to remain hydrated, minimize water use, and their need to undergo photosynthesis and capture CO2 out of the air. And so one of the most important set of open questions in plant science and in the future of managing plants for agriculture is around that, how to optimize that management, the compromise between carbon dioxide and water. And just briefly, it's an open story. And for example, the impact of rising CO2 on plant life, whether it is actually a benefit to plants because they have more accessible CO2, or whether it leads to unexpected outcomes is still unknown. The simple-minded answer is that more CO2 is better for plants. They have to open themselves up less to feed photosynthesis than with less CO2. But that is not empirically entirely obvious yet, given that even with these quite dramatic changes in CO2 levels that we've experienced. So another good question.
Okay, thank you so much, Professor. I think we can continue forward.
Thank you. All right, good questions. Keep them coming. So I am so this idea of tension in plants has been documented using that that manual, rather painful tool. And what we see is that indeed it is the mangroves that here day and night experience large stresses. It's the arid climate plants that experience the largest stresses, whereas actually gravity and the largest plants really only experience some small multiple of rho GH, of the pressure fighting gravity to maintain their stress. So where we entered this problem was to ask, is this really true? We actually had that question. Is it really true that plants use liquids, metastable liquids under tension? And so 12 years ago or so, my first graduate student set about to build a synthetic plant, which was essentially a microfluidic structure inside of a relatively simple hydrogel. And so these are the microfluidics of vessels that are visible, that are like the macroscopic scale vessels in a plant. The nanoscopic structures, the nanopores, are provided by the hydrogel material. Well, it turned out that we could actually, with this little synthetic plant, this, you can think of this as the roots and this is the leaf, we achieved stable stresses well beyond what had been documented in plant life, and in an important sense, did a proof by construction of the viability of this way of holding on to liquids in the materials that are available, and in this case, even in organic materials that are available for plant life and for technology. Interestingly, 8 years later, a group of plant scientists in Australia found very much this same limiting stress in this Wartyzeria from the outback in Australia, suggesting that perhaps there is something special about this stress that we've found. We've now found this is our limiting stress in a variety of different devices that we build that put liquid in equilibrium with the dry world and under tension. Now, we do not have any excuse for this being the limiting stress at which water breaks or boils. If you think the actual calculated spinodal of liquid water, like I said, is expected to be beyond 1,000 atmospheres. But there is perhaps an impurity associated or a catalytic site for the creation of vapor bubbles in liquid waters under tension that is shared between synthetic materials like ours and plant life. But in any case, 200 atmospheres is a lot of pressure to work with. And so we have looked to using this stress to explore various scenarios and to build technologies. So over the last 12 plus years, we've taken this concept, a plant-inspired concept, and turned it into various formats, including a technology that I will tell you about more. Very briefly, by building what are essentially micro-nanofluidic systems, we've been able to look at some of how the architecture of plants is the biophysical basis for the architecture of plants and how it controls dynamics in the presence of phase behavior and the general thermodynamics. We use now a porous silicon that gives us a few nanometer scale pores. And since we can create, with this drying stress, we can create hundreds of atmospheres of stress, negative stress, we can start to move liquids through tiny nano confinement and understand how nanoscale transport occurs. This is dynamics. What we're watching here is little boiling events in pockets of bulk macroscopic liquid being put under stress. And we can look at the thermodynamics, for example, of mixtures. Interestingly, the solubility of oxygen in water goes up by a factor of 2 as water gets put under tension within scenarios like this. But my main story is going to be about this most matured version of our synthetic plant, which is really a stress meter. And so what we've built is we have xylem-like vessels connected to nanoporous silicon that connect back to a bulk liquid for which we can understand the thermodynamics very easily. And this cavity of bulk liquid is connected to a microelectromechanical pressure sensor. And so we bend silicon, and we can understand the state of stress of this liquid, and that gives us information about the outside world. So coming back to our minimalist representation of what a plant in water is, we now have a plant that has a closed end where we have a bulk pocket of liquid water. We allow it to come to equilibrium with samples of interest, where this is essentially a measure of the chemical potential. And then we can measure this liquid pressure as a simple voltage on what is a bridge circuit on our pressure gauge. And that means we can turn, this is the digitization of inside state of a plant in a minute. We take this chemical potential of the sample and turn it into a simple voltage. So this is a device that we can use. It's a technology. Also, it's essentially an osmometer in some circumstances. It's important that we understand its transient behavior. It's got minute scale transients. We can get a calibration. We calibrate its the output, the voltage versus the imposed chemical potential or water potential. And so this is now a maturing tool that's even actually licensed, patented and licensed to a former graduate student of mine in a company. And what I'm going to tell you about is how this is a step towards the Internet of living things, where we can take a device, embed it directly into the tissue of a plant, and allow for local equilibrium to occur between the water in the tissue and the water inside of our device. And then that becomes an electrical signal that we can digitize and send out into the cloud and into our models. And so the company is Floor Pulse that has brought this and now is based in California. And so I'm gonna tell you about work. I'm going to use the example of almond, but this is now working in a variety of different crop species. So in almond, we've worked with a fellow named John Monroe, who owns an orchard in Northern California for almonds. And this is a tree crop. And he's owned this farm through what was a devastating drought in Northern California, and understands and invests in the question of how to manage water more efficiently. And so he has this unit operation that he wants to control. He has to understand how this plant is using water so he can make decisions about how to irrigate it. And just for reference, the Central Valley of California is really almost desert conditions where they have access to water, and so they can do agriculture if they give the right amount of water. So a grower like John Monroe has to worry about the complexities of the atmosphere, the complexities of the biology, making decisions about the compromise between carbon dioxide and water. And of course, there's all the below ground, which a mature almond tree will have potentially hundreds of kilometers of roots living invisibly under the ground. And so this is where we like to gain this internal information about the system. So Monroe does invest, has been investing in using this painful manual tool a couple times a week at most to measure the stress. And so here I'm reporting the water potential in atmospheres of stress or bars of stress, negative. And the trouble is, you just really don't see much by taking this type of point data on this living organism interacting with the environment. So this he gets this kind of advice from a service provider on the internet when he puts his data into the web. He has high stress, he has low stress, sweet spot, but it's really hard to see what the full dynamics is of this living unit operation. And he makes this decision about irrigation basically on the frequency that he has information. So this was one of the first installations that my former student Michael Santiago did when he started out in California. And it was, most importantly to us, it was we basically could connect the dots. The tool that John Monroe had been using, the manual tool, is known to be accurate. And what we saw was much more dynamic information, but importantly, connecting the dots of the conventional method. So this starts to show that John Monroe is actually spending a lot of time outside of an optimal regime in terms of management of his plant. But more interestingly, it starts to show the full life, the pulse of a plant. And what we see here day and night is that we see the dynamics associated with the internal plant control. This is the little valve in the leaf of a plant, of which there are millions, that is opening to allow CO2 to come in. But for each CO2 that comes in, hundreds of waters go out. And so here in the light bands, each day water is being lost and the system is being put under stress. And then at night it closes and relaxes back towards a minimum stress, a maximum internal pressure, and again all day long. So already we're starting to be able to see what the dynamics of this system are under the biological and environmental control. Now we're also seeing on this longer time scale the dynamics imposed by the management of irrigation. And so we have two important time scales here. One, the driving of the biology by the daily rise of the sun, and the other, the driving of what is essentially through the soil of the week-long scale periodicity. What we'd like to do is, now that we have continuous information about this plant, it's always in some sort of transient, it's to extract out some information that goes beyond, that gives us information about the invisible physiology of the organism. And so this now allows us to start to look at this organism and its coupling to the environment. What a goal would be, an important goal would be to take the things that we can get from the local weather station, the radiation, vapor pressure deficit is a measure of the relative humidity. The wind speed controls the boundary layer interactions with the leaves, and simply the temperature controls all of the thermodynamics of the system. And we'd like to go from this to an ability to predict this. And to do this, we need to bring in models that are essentially transport models of the different components of the system, the complex porous, unsaturated porous medium of the soil, the tissue properties, and the boundary layer properties of the outside world, and the biology of this control valve in the system. And we want to, how much can we extract out of information by having a point probe in the center of this system? So it turns out we're going to represent all of this information about thermodynamics and transport in essentially in circuit-like diagrams like this, where we have capacitances and resistances, and those resistances and capacitances are controlled by the detailed biology, mechanics, and thermodynamics of the system. So just to give you a sense of how we can start to think about this, one nice little experiment that is essentially run every day is that here we're showing the evapotranspiration, the amount of water lost by the plant through the day from 5 in the morning to 8 at night, and then nighttime, basically it shuts off. And here's what that's the driver of dynamics. Here is the response of the plant, going from a modest 10 atmospheres of stress down to 18, and then relaxing back at night. So if I drew that circuit, the simplest version of a circuit for the plant is a single hydraulic capacitance being fed through a single hydraulic resistance. And this is soil, plant, atmosphere continuum for these two. So what if we just say that this is a current source driving a simple RC circuit like you saw in high school or freshman physics? Well, this is a solvable, analytical solvable system. It tells us that we should have a single exponential relaxation every night as the system turns off. And so this is, and you can see that it does seem to be a kind of saturating exponential relaxation each night. So we can go through and find out a first parameter, which is the characteristic time of this system, which for an RC circuit is R times C, and by fitting every one of these relaxations each night. And indeed, we come out with an important information about our unit operation, that it has a characteristic relaxation time of about 5 hours. This also, each night, we watch the system relax back and fail to equilibrate with a potential, which is the potential in the soil around every bit of this 100 kilometers of route. And we can get out another piece of state information, which is the effective state of the soil that the plant is experiencing. So this is how we can use what is just a point measurement within the plant to start to infer information about its dynamical properties and its state, even the state of parts of the system like the soil that we cannot see. So it turns out to capture the full dynamics, we need to actually put in more than one resistor and capacitor. We need to put in what the most complicated physics is in the soil, in the soil interface with the plant. And we can start to do quite well mechanistically, fitting the behavior both on a daily basis and through the cycles of irrigation. Where we win is that we get predictions of things that we cannot see. And one of the important insights here is that we see the prediction for the variation of stress within the bulks of the soil. And this is where many people are tempted to try to track the status of water in a plant system. And what you see is there's almost no dynamics in the soil, even as the top of the plant goes through huge variations on a daily and weekly basis. And so this is where the foundations of building out accurate predictive models of plant systems, and the basis to start to treat plants like unit operations in a manufacturing process. Here, teaming up with Funshi Yu, a process systems engineer in my department, to look at things like model predictive control, where we could tell John Monroe, make an optimal decision for today based not just on the past information, but based importantly on weather predictions into the future, economics, environment, and crucially our process model, which I've just given you a sense of how we build. So this is the beginning, we hope, of Industry 4.0 out into agriculture. So I'm going to finish soon and just give you a summary here of the kind of things that we need to do. Well, because I've just told you how we treat a single organism, a single plant. We need to turn this into a distributed model. And they're interesting questions, again, coupling closely to the types of expertise that we have as engineers about how what kind of heterogeneity we expect across a population of plants. One source of heterogeneity that we're exploring is due to the boundary layer, that at the edge of this field, we will have plants experiencing the leading edge of a boundary layer, and deep into the system, we will have different behavior. So understanding that biological and environmental heterogeneity. We're interested in bringing this out in natural context. We have a collaboration getting similar types data from the very tops of redwoods. Interestingly, the tops of redwoods have dynamics that look a lot like the trees that we study that are of totally different scale. Although you'll see that every night the redwood never goes back towards zero. It's stuck at, this is a measurement at about 100 meters, and it never returns beyond the rho GH of about 10 atmospheres. And then we have an opportunity to go after the biology of this biological control circuit, these stomates, and they are really like a PID system. And on some occasions, when we shock a plant, we actually send it into an instability, a ringing of our control, of the internal control circuit, that start to give us hints about what the internal control parameters are on the biology, on this critical biological process of navigating the compromise of CO2 and water. So I'll just tell you very briefly about a project ongoing right now where we're bringing sensing even deeper into this boundary layer on our atmosphere, inside leaves, with a dispersible sensor of this same parameter water potential based on the dynamics coupling of a gel and a fluorophores. And what this allows us to do is take an image of a leaf that would look like this without our sensor and give it life. And what you start to see is that the simple, what you think is simple behavior of how the leaf interacts with the atmosphere is actually hugely dynamic as these the stomatal control is coupled to changes in the atmosphere and internally coupled, and in this case showing a rather chaotic-like dynamics of the water stress here imaged spatially and in time. So with this type of tool, where we'd like to go with our vision of Internet of Living Things is that we'd be able to take this internal state variable of stress within the leaves of plants and have it become directly visible as an optical image from a drone or even perhaps satellite, so that we can track the behavior of this subtle regulation of water status and CO2 over large populations, feeding, for example, the ability to select water efficient plants. So I made it to the end without Kriti stopping me again. So I'm certainly happy to stay on and have questions for as many questions as you have. Thank you very much for taking the time this morning or this evening on your side of the earth for this talk.
Thank you so much, Professor. So I think we have quite a few interesting questions. I'll take up some of them in the interest of time. So the first one is, can you brief us about the relation between increased water usage efficiency, the tree growth, and its impacts on global climate?
Water use efficiency is focused around the control of these valves in the leaves, the stomates. But it's actually a function of many characteristics of the plant and in the context of agriculture of management. And so today, agriculture, as some of you may have some experience, is a hard business, and agriculture tends to be very conservative. You want to keep your plants alive first and foremost, and so a human behavior is one of the biggest challenges. Managing a human behavior is one of the biggest challenges in water efficiency. People put more water on their plant than is necessary, often by at least a factor of 2, because they're scared that they could lose the plants to drought stress. So this is a place at the simplest level, before creating closed loop control of water management, is to simply be able to give a farmer or a grower more confidence about the state of stress within a plant, so that that person can be more conservative, be more conserving of water, and do it with confidence. But now, at the scientific level, there is an important question of how to push plants towards more efficient use of water for a given amount of CO2. And I'll tell you, one of the things we're excited about with this new tool is so this is a detailed view of a stomate and the exchange of CO2 and water. Today, the thinking in biology is that everything is controlled by this valve, and in an important that the internal surfaces of the leaf remain extraordinarily very near saturation. Even though the liquid stress is large, the relative humidity is very near saturation. we and others start to have evidence that some plants, under some conditions, allow this internal boundary condition on the vapor to go much more unsaturated. And if that is true, there may be a biology, there may be genetic ways to get plants to have tool beyond the stomate. So if the plant could allow the internal relative humidity here, EI, to drop, it could keep the stomate open, collecting CO2, and still limit the loss of water. And so that's a future looking answer to say that we're hoping to be able to find traits that allow plants to be more water use efficient by allowing them to control water loss without impacting CO2 uptake, and to manage this so-called gamble for life between CO2 and water. So good question. Sorry for the maybe excessively complicated answer.
Thank you so much, Professor. So I think one of the similar questions is, it's not exactly similar. So you talked about water usage efficiency in drought-prone areas. Maybe it could be a very big application. But is there something to control production in the rainy areas, the areas which are full of rain and lots of water?
So that is a good question. In fact, I live in an area that is a humid, non-arid area, and there's other parts of plant biology. So, for example, rice. The agricultural strategy for rice to have it live an entirely flooded existence during part of its growth is based on a special biology of rice that allows it to maintain oxygen transport into its roots, even in the presence of essentially stagnant water layer. So there's a whole nother biology and transport phenomena. In that case, transporters of oxygen down in that keep from anoxia or hypoxia killing the plant, which is what would happen to other plant species. So the tools that I've described here are not easily applied to the case of excess water. I'll just say the experience here in the Northeast of the United States is that although on average, historically, we have enough water, we now, with increasing frequency, have extended unpredictable midsummer droughts that can completely devastate the production of crops in our area. And so while nobody had irrigation installed in our region until recently, now they're putting it in, and in fact, relative to California, it's a much more as these less predictable droughts occur, it's a stronger demand for the type of model predictive control that I described. In California, you can be guaranteed it's going to be dry all summer long. You're working in a desert essentially. In New York State, you may go from excessive rains to complete drought, and to have some ability to respond to that wisely is a high priority. So it's a good question. Water, it can be trouble on both ends, too much or too little.
Thank you so much, Professor. So I think we'll take up last 2 or 3 questions. So one of it is, does the quality of water, that is the concentration, pH, etc., does it affect the water stress?
Yeah. So I didn't tell you about root biology in any detail. Roots, relative to my minimal plant. My minimal plant is this minimal plant. This is not a good representation of the root. The root can hold on to water in this manner relative to dryness in the outside world, but the root has a living membrane, and I see somewhere else in the questions about aquaporins. So water mostly gets into the vessels within the root through membrane processes and aquaporins. Those same, and I think the question about aquaporins related to building membranes out of them. So aquaporins are extraordinarily selective for water, and that means that they're in some sense the ideal and an ideal limit of reverse osmosis. So plants can be quite robust against solutes in the water within the soil. If those solutes are at sufficient concentration, like the mangrove in ocean water, it will shift the thermodynamics and create more stress within the plant. So ocean water should give you huge access to water, but only if you can overcome the 35 atmospheres of osmotic potential within the ocean water. Yes, there is a sensitivity that starts to be very specific and biological to pH, specific solutes, toxins, but in general, what plants are doing is essentially what I would call a reverse osmosis. They are using negative pressure, reduced pressure inside the tissue to pull liquid through what is essentially a reverse osmosis membrane. And in that process, they are able to select out solutes in a way that is biologically selective. So good question.
Okay, thank you so much, Professor. So I think you already saw the question about aquaporin, about how it can be used for filtration process like membrane separations.
Yeah. Well, it is in an important sense. It is the ultimate pore. In being extraordinarily selective for the passage of water. So it's not an area that I know well. It does present real challenges in terms of an engineering context, because it is a transmembrane protein. It needs an intact membrane. Plants themselves, of course, do bioremediation. Plants can help clean up soils. And for now, that is the best way to have a aquaporin deployed in the environment, is to keep it associated with a plant, because of course the plant will continue to renew it, maintain it. But I do think it's a worthwhile target, like here in terms of bio-inspired engineering, to go after membrane structures, which today our membranes are very primitive, hugely primitive compared to biological membranes. To go after that type of selectivity, and as I understand, quite good permeability, to get to truly ideal osmotic membranes and membranes for other uses. But we have to be respectful of the full, aquaporins emerge on the surface of a living cell with the full machinery of a eukaryotic cell that has gene regulation, continuous renewal of protein, maintenance of that host bilayer membrane. But it would be a great target as a protein engineer to try to work towards a stable semi-synthetic deployment of aquaporins for technological uses. So keep it in mind.
Thank you so much, Professor. I think in interest of time, we will not be taking up any more questions. But it was a really insightful session, and I personally got to know a lot from the session. I learned a lot. So thank you so much, Professor. So I would like to invite Manan Kasana, who is the overall coordinator of azeotropy, to present the concluding note.
Thank you. Thank you all.
Yeah, thank you, Preeti. So hello everyone, I'm Manan, overall coordinator of azeotropy 2021. So first of all, I would like to thank Professor Abraham on behalf of our team for taking out the time for this event and providing all of us with wonderful insights on the applications of chemical engineering technology, like on plant life. So it was pleasure hosting, and the topic was also like very interesting, something that we don't learn in our regular undergraduate chemical curriculum. So I'm sure everyone also found it interesting, just like me. So thank you. I would like to thank all the participants as well for attending the session. I'm sure that curiosity has been picked for all the participants, and everyone enjoyed the session. Thank you so much.
Thank you all, and best
Thank
of
you
luck.
so much, Professor.
Yeah,
Thank
and if you would like, if anyone would like to follow up on this odd topic in chemical engineering, please be in touch. Okay.
Surely,
Sure.
Professor. Thank
Be
you.
well.
Bye-bye.
Okay. Bye-bye.
Bye-bye.